Hey everybody, welcome back to yet another Journal Club brought to you by Lifespan.io. And I'm here with the regular crew. And this is the last Journal Club of the year, officially, I believe, unless we have some sort of snap emergency Journal Club, which I doubt. So we're gonna take a break. So we got something a little bit special planned today. I think it's a nice, tight, short paper, and it's something we haven't really touched upon. We've talked about it, but it's the big white elephant in the room, COVID-19, right? SARS-CoV-2. We are approaching year 2022. Geez, it's been going on that long, the year of our pandemic. We are about 1,000 days into 2 weeks to flatten the curve, folks, and here we are. So it's still going. And so what is COVID-19 and what does SARS-CoV-2 have to do with aging? I mean, this is just two diametrically opposed things, right? We've got a contagious illness, a virus, and then we have something very complex happening in every living system, but we focus on human aging and human longevity. Well, as most of us here probably know or realize, COVID-19 strikes the elderly disproportionate to pretty much any other demographic. So obviously, if you have health conditions, it's going to strike you pretty hard. It hits lung tissue pretty bad. But the number one driver for basically mortality for if you catch COVID-19 is age. Age is the biggest risk factor. And if you look at the CDC, and I don't have the stats right here, but you can pull them up. If you're relatively young, if you're a child, I'm not going to say it doesn't affect you, but it's pretty negligible, the numbers. But then when you start looking at people that, once you start getting past the age of 50 and 60, especially, and 70, the numbers really, really skyrocket dramatically. So it is definitely affects the aged disproportionately. And one could argue that, if we had a much more younger demographic out there, at least biologically speaking, if we were biologically more youthful, it's debatable whether this pandemic would have even, been really noticed if it wasn't for the fact that, all of our demographics are skewing towards the aged. And thus, we're seeing a lot of deaths, unfortunately, in that demographic. So with that, the question is, why is COVID-19 impacting the aged so disproportionately? And of course, we can argue, and rightly so, that a lot of systems, as we get older, basically become less effective, less efficient, particularly the immune system, right? So for your acquired immune system to be active, your cells need to replicate pretty readily. So you get memory cells forming. So if you've been been infected with some antigen, such as COVID-19, your body needs to mount a immune response. And then if you're going to be vaccinated, you also need to have your cells replicate and basically last as memory cells, memory T cells and B cells, to mount a strong secondary response. And all of those systems get suppressed, go down as we get older, right? So cellular turnover goes down. So the immune system becomes less efficient. So, it's understandable then, just from that perspective, why immunizations would be less effective in the elderly, and also why illnesses that are infectious would strike the elderly. But there's another reason that was teased apart in this paper that is another reason given why the elderly may also be, disproportionately affected. And it's the first paper of its kind that links telomere erosion to the ability for COVID-19 to preferentially infect cells of folks and, who have shorter telomeres. And folks who have shorter telomeres tend to be older. So let's take a look at this paper. So let's take a look. So first, before we take a look at this paper, let me pull up a little PowerPoint presentation here. Slideshow, slide. Okay, oops. Let me share my screen. So pardon me, I didn't have time to get all the ornaments for my Christmas tree. Found this on Amazon.com. So those of you wanting to wish a Merry Christmas and a good riddance to past years, there you go. So that's all the decorations I have for you folks. Last minute. So very quick synopsis of what SARS-CoV-2 is doing. There's tons in the literature right now, right? So for obvious reasons, but it's a virus. It's an RNA virus. And really for the purposes of this paper, it docks to these receptors called ACE2 receptors, angiotensin converting enzyme 2 receptors that are found in a number of different cells, but are found in cells in specific cells, primarily in the lung epithelia, right? So this is where this virus is going to infect, and it docks. So viruses basically, they dock to receptors. HIV receptor it docks to, receptors on CD4 cells and the CCR5 co-receptor. So every virus has its kind of repertoire of receptors it docks to, and SARS-CoV-2, the causative agent of COVID-19, docks to these ACE2 receptors. And this is, if you want to learn more about them, here's a paper. ACE2, much more than just a receptor for CoV-2. It's not just a gateway for SARS to come in, but it performs a vital role, right? So viruses basically hijack normal systems, operating systems to get in. So there's a potent signaling molecule, angiotensin, which is required to regulate blood pressure and does a lot of other things in the body. And it gets basically broken down and degraded by these ACE2 receptors. So the bottom line is, before this paper that we're going to jump into is published, it was noted that ACE2 receptors go up in people in the elderly. So ACE2 receptors are higher, and basically at higher levels. And that may be a sort of a homeostatic beneficial thing, because if you have too much angiotensin that isn't basically converted by the ACE2 receptors, it leads to inflammatory conditions, right? So ACE2 levels going up might be basically a beneficial response to elevated angiotensin levels in the elderly. But unfortunately, the virus likes to dock to ACE2 receptors. So that, of course, is a problem. So stop sharing here. So that's just basically a quick synopsis. So I'm going to jump into the paper in about 30 seconds, sip my coffee if anybody has a question about SARS-CoV-2.
Yeah, what sort of coffee are you drinking?
This...
I'm just messing about.
Well,
actually, yeah. No, I am curious now. What sort of coffee are you drinking?
Well, it's not Folgers Crystals. It is coffee that Gene and I pick up at a small coffee shop in Lower East Side of Manhattan, right by Cooper Union, called Puerto Rico, spelled Puerto Rico, P-O-R-T-O. And it is, we usually get, or she likes to get, the least acidic beans, and we grind them ourselves, and we use a French press. And this is the coffee I'm having. So quality coffee.
Well, I'm just embarrassed. I use instant coffee.
Oh, yeah.
Hey, it's Dow Egberts.
I mean, come on, Steve. You're not in prison anymore.
Not anymore. And anyway, it was never, the allegations were never proven. So anyway, coffee talk aside, I don't have any questions about COVID. I'm kind of sick of hearing about it, to be honest. But it is interesting that we're talking about it today in the context of aging, which is a little bit of a flip and actually makes COVID a bit more interesting for me because, well, I'm a little bit done with it.
I have a question. On your cartoon, there were two kinds of ACE receptors, and it looked like the virus was only going into one of those two kinds. Is that correct?
Yeah, the ACE2 receptor. Just the angiotensin converting enzyme 2 receptor. I'll be honest with you, I haven't really dived into the literature too deeply, so maybe there's some other thing that it docks to, but everything you hear about is the ACE2 receptor. I don't know if there's a co-receptor or anything, but definitely the ACE2 receptor. Questionable if something else. So, okay. So, so obviously that opens up, the idea of blocking the ACE2 receptor somehow, or coming up with drugs, or getting down regulating it. Now, that being said, we're going to dive into this paper in a moment. The ACE2 receptor is performing a beneficial function, so you clearly don't want to just obliterate the ACE2 receptor. If you do something like that, you might want to do it temporarily and then stop that, because it's detrimental. You might have elevated blood pressure, all kinds of other issues, because it's important for regulating a lot of functions, a lot of renal functions, I believe. So, but anyway, let's get to the paper. So here we go. So one thing I like about this paper, and I mentioned this to Keith earlier, is it doesn't have a lot of data. And I mean that in a good way. I mean that in like, they could have kept going on and on and on, and basically, then doing epidemiological study and looking at, the telomere lengths of a population of like 10,000 and going on and on. But they left it at 5 figures. They basically, kind of connected the dots to a telomere erosion, and specifically the DNA damage response that causes the ACE2 receptor to upregulate, and said, wow, this looks promising. And I like that, because, the data gets out there quickly, and somebody else can do follow-ups, right? So when I say that there's not a lot of data, there's not a lot of follow-up data. There's a lot of data that basically is already connecting the dots here, which is great. So I meant that in a positive way. Sometimes papers definitely suffer from too much data, and things get kind of at the periphery all over the place. But this one kept it tight, and it was pretty good. So this is I don't know how I'm pronouncing this. Seppi et al. Sarah Seppi et al. This is mainly a lot of labs from Italy, along with Jerry Shea, who basically very famous in the telomere field, and he's over at UT Southwestern Medical Center now. And a lot of the other groups are from the Molecular Oncology Foundation, Milan, Italy, Palermo, Italy, University of Palermo, and Sapienza University, Rome, Italy, and Istituto di Genetica Molecolare at Pavia, Italy. I just butchered that accent. Okay, anyway. By the way, as an aside, if you'd like to see an amusing movie that's got a great plot, go see Lorenzo's Oil. It came out back in the 90s. It deals with a father and trying to look for a cure for his son's illness, and he connects with a chemist that basically tries to synthesize this molecule and succeeds, and he basically goes around the entire FDA pharmaceutical industry to look for a cure for his son. And it's a very uplifting story. But I say that because who's the actor? Steve, you got to help me out. God, forgot. Not Trading Places. Lorenzo's Oil. Who's the main actor? Anyway,
to be honest.
I was about to say, you could Google it. Worst Italian accent ever. They couldn't have picked, absolutely awful. And
would that be Nick Nolte?
Nick Nolte. Can you imagine
Just
imagine Nick Nolte with his gravelly Midwestern voice. I don't know where his regional accent is. Nick Nolte doing an Italian accent, a thick Italian accent through the movie. That's all I gotta say. Anyway. Apologies to all of Italy if I just insulted an entire country. Okay, so it's end of the year. I'm getting a little relaxed here. And it is just coffee I'm having. So the paper is DNA damage response at telomeres boosts the transcription of SARS-CoV-2 receptor ACE2 during aging. So that's basically the punchline. So it's interesting here. We know that they've known that SARS-CoV-2 receptor ACE2, the angiotensin converting enzyme 2 receptor, goes up, but it wasn't clear why it goes up. And we know that obviously the virus docks to it. And if it goes up in aged individuals, well, it'd be kind of important to tease out the pathway. And what they do is they notice that this is due to telomere erosion, and not just due to shortened telomeres, but to the signaling that happens at the telomeres. So let's take a look at some of this data here that they got here. So this is figure 1, and let's see, is the legend above or here it is. So they do some background and stuff, right? ACE2 expression increases during aging in mouse and human lungs. So they look at young mice and they look at old mice, so 2 to 3 month old mice and 22 to 24 old mice. They look at messenger RNA levels, so they do RTQ, PCR, and then they look at do immunohistochemical staining in lungs to look at protein levels. And then they look at, what do they look at here? So these are samples that they received, well, it's in the materials and methods where they received these samples, but again, immunohistochemical staining of lung parenchyma of young people, 20 to 35, and old, 60 to 80 years old. Apologies to anybody who's 60 and up, they're referred to as old here. So that's their designation. So according to this group, you are old officially. So ACE2 messenger RNA levels, well, there they go. So they get higher. So these are in mice, right? So young, old, relative levels, young mice, old mice, immunohistochemistry. So ACE2 levels go up, both in mice and in humans. So it's pretty conserved, mice and men, right? So here are in human samples, young, old, right? So these are specifically in cells that are, so these are alveolar, what are they basically? So these are these type 2 pneumocytic and CD31 positive endothelia in lungs. So these lung specific cells that are known to basically have higher levels of ACE2. So ACE2, I mentioned, is found in a lot of different cells, but it tends to kind of really be at a much higher level in certain types of cell types inside the lung tissue. And these are the ones that they're looking at here. So those levels go up in those cell types. So old, young, so this is statistically significant. ACE2 levels in AT2 cells, so those are the ones, and also in endothelial cells, right? And this happens in humans as well. So these are the human, these TTF1 and CD31 positive cells are the mouse equivalents that they're looking at in humans. And this is basically, what is this? This is, they're showing here, this is transcriptomic data. They're basically showing that this ACE2 expression level goes up preferentially when they look at many different cell types. It's kind of a weird plot, but basically it clusters in these cells that are type 2 pneumocytes. And that's what that's demonstrating. So levels go up in old mice and in old human samples. Now they look at what happens in cells that have shortened telomeres. So, you have older cells, but let's look at cells that specifically have telomere shortening. So they have, these are cell lines. Well, not cell lines. These are normal human fibroblasts, right? That basically don't express telomerase. So they basically can only be passaged so many times until they hit the Hayflick limit. So early passages, population doubling 34 to 37. Late passage, population doubling 61 to 64. They look at the normal fibroblasts, and they also look at later human bronchial epithelial cells. And then they look, and then they also, and I'm going through the figure legend because I don't want to bounce back up and down, but I'll get to the figures. And then they look at mouse representative cells with shortened telomeres. And in the mice, what they do is they do these generation 3 telomerase, these TERK minus homozygotic mice that basically, so mice have really long telomeres. So to get the telomeres really short, you got to not passage them, you got to breed them several generations, and then the telomeres get shorter and shorter. And then the mice die. So they basically have these generation 3 TERK minus. So I believe that's the RNA subunit of telomerase that's being knocked out. And they looked at the ACE2 mRNA levels and the ACE2 protein levels. And they also look at the ACE2 protein levels in, again, type 2 pneumocytic and CD31 positive endothelia and cells from these TERK mice. So anyway, I mean, it's pretty clear that ACE2 levels go up. So messenger RNA goes up, protein levels go up. So here's so two different cell types. These are human passage cells here. These are the ones that are being from early passage and late passage that I just mentioned, and then immunohistochemistry of the mice. So wild type G3 generation 3 Turk minus. Again, levels of ACE2 go up. And the same thing with the specific cell types that they're looking at with staining the cells, these AT2 cells in the alveoli. So these are within the lungs. So again, ACE2 intensity goes up. So I'm gonna stop sharing here. So any questions?
So I thought figure 1 was actually really impressive. To the best of my knowledge, this is the first reports that strongly show ACE2 expression like rising in adults. In the intro, they cite the literature as showing like, all right, there's some papers showing that there's more ACE2 with age in humans. But the existing data seems to all be like kids versus adults. And when you're thinking about COVID-19 risk, where there's a very strong component later in life, or risk component later in life, I think this is the first data that backs that up by looking at ACE2, by actually doing the comparison to like 60 to 80-year-olds versus like whatever their young group is, 20 and 30-year-olds.
Yeah.
It's also nice that they correlate their RNA data to protein data with their immunostaining. Because like it's really easy to just say, there's more mRNA, but they actually go further than that. So yeah, that's pretty cool.
Yeah, I mean, data is like really clean. I liked it. It's just very very logical.
I'll throw in one other comment for figure 2. So they grow out their cells for a real long time. Their late passage on their epithelial cells, which I think are like bronchial cells, is allegedly like population doubling 140. Like these guys have been in culture for a while, and they attribute this to it's probably telomere shortening, and that might be true. But like when you grow cells that long, like stuff happens. Like they could be aneuploidic. They could have picked up telomerase overexpression. They could have picked up like some G1S checkpoint override where they just like start steamrolling through mutations. So
yeah, well, what they show, that's a good point. And what they show later, I think they use ionizing. I don't know if they show the data with the ionizing radiation, but basically, it's not just so telomere erosion. They note will basically trigger a DNA damage response that causes upregulation of ACE2, and they show later that this is specifically by upregulating transcriptional factors that basically switch on ACE2 right at the promoter using a simple plasmid system with luciferase. But they note that any kind of DNA damage and the subsequent DNA damage response will trigger ACE2 upregulation. So yeah, I guess, you could also argue that, if you get those cells in culture, like all kinds of other stuff can happen and that will trigger DNA damage response. But I guess to their point, telomere erosion too will do that.
Yeah, and also to their credit, they test two different types of cells that are fairly dissimilar, and I think that increases my confidence in their findings quite a bit.
So okay, what do they do here? So murine embryonic fibroblasts. So this is a system where they have, so TRF is, I think that's, what is that, part of the shelterin complex that basically protects the telomere ends from being recognized as a DNA damage. So they have a couple of systems here. I'm just going to remind myself. So they have a conditional knockout mice. Yep, shelterin complex. So they had TRF2 conditional knockout mouse embryonic fibroblasts, these MEFs TRF-FF carrying a Cre recombinase inducible by 4-hydroxytamoxifen. So basically, they add 4-hydroxytamoxifen, and whammo, the TRF2 gets basically excised out, and you lose the sheltering complex component, and you have basically the telomeres are now exposed and recognized as a DNA damage. And then they had HeLa cells with a doxycycline regulated expression of a short hairpin. So this is RNA that basically inhibits the messenger RNA against TRF2 to knock down TRF2 expression. So I guess these systems were already extant in these different cell types. So they were able to test it in, like to your point, Michael, 2 different cell types. HeLa cells, human cells, and also mouse cells, right? So, and HeLa cells, we can also argue those are really whacked out. Those are, but they had normal cells that were undergoing passaging, and then now they have HeLa cells. So it's interesting that we'll be seeing all of this kind of all over the place. Okay, so so they'll note that the TRF2 knockout or knockdown, either of those systems, they'll lead to telomeric DDR activation, as shown by increased levels of gamma H2AX, which is basically a histone that basically increases in response to telomeric damage. And also these at the location of telomeres, you get these induced long non-coding RNAs, TDILNC RNAs. It's the longest prefix to an RNA I've seen, TDILNC RNA. So induced long non-coding RNA, which basically, I'm not familiar with that pathway, but it seems to be required for signaling downstream for the DNA damage response. Okay, so let's go to figure 3, which is down here. Okay, so so here are your murine embryonic fibroblasts that basically have been treated with, I forgot what, tamoxifen, sort of variant of tamoxifen to Cre recombinase, and basically get rid of the sheltering complex. And you have these punctate foci of gamma H2AX forming at the telomere ends. And these are days after this variant of tamoxifen treatment. So you basically have a loss of the sheltering complex. So these telomeres are not, I think their point is I don't know if they measure the telomeres here, but they're not, so it's not the telomere erosion per se that's triggering this, but basically the exposure of telomeres, and that they'll note later the consequent damage response. So ACE2 levels, messenger RNA levels go up, right, in these cells. And then the same thing is seen in these HeLa cells that have these short hairpins that knock down TRF2, increased gamma H2AX, and you have increased ACE2 relative messenger RNA levels after doxycycline treatment. Next, what they do is they now do this in, I believe, in mice, in vitro. So 53BP1, I believe it's, what is it? Part of the complex that recruits P53, I believe, to DNA damage of foci. And let's see what, so obviously ACE2 levels also go up at tamoxifen treatment, but I just want to double check here and see where in the mouse life cycle this was. Inducible TRF2 knockout mouse, which following tamoxifen TRF expression leading to DDR activation, telomere accumulation foci 53BP1. Okay, so they notice, so this is the ACE2 levels they notice are in the liver. Are those liver cells that they're looking at here, the mice? So I'm not sure exactly how many days this is after. So these are in mice. So let's see. Yeah, okay, liver. So these are liver cells, and then QRT-PCR, the ACE2 messenger RNA levels going up. in these mice. Okay, so if we lose this part of the sheltering complex, minus any potential telomere attrition, you basically get an increase in ACE2 levels. So I'm going to stop sharing here. Questions?
Yeah, tamoxifen seems to regulate estrogen receptor. And does it also regulate the ACE2 receptor?
Good question. I don't know, and I'm not sure if they had that control. That would be a good control to have, and then basically add. Let's take a look. I didn't see that control. They had vehicle and tamoxifen, but they did not have tamoxifen in a non-transgenic mouse. So, good question. Anybody else want to comment on that? I'm not, yeah. I'm not, you know, maybe it could. So we just pointed out a control there that we might utilize in a follow-up.
I mean, it seems unlikely, but yeah, maybe.
Yeah. Okay, we'll shelve that temporarily. We'll got 2 more figures to look at here. So here they're going to take a look at how DDR activation upregulates ACE2 gene, and it does it transcriptionally. So they do a gene enrichment analysis to look at all the signaling pathways that are upregulated so this is with the DNA damage response. Let's take a look here. So this is where they do the very simple and to the point transfection with a ACE2 promoter that basically turns on luciferase. So they look at gene enrichment analysis as the top 100 transcription factors, but that's in table EV1. We're not looking at that here. So they basically look at, you know, all the potential transcription factors that are upregulated that might basically they're also, a lot of them have binding motifs that are enriched at the ACE2 or enriched at the ACE2 promoter level, at the ACE2 promoter locus. So that led them obviously to speculate that this DNA damage response, along with other pathways that get upregulated, probably integrate at the promoter. So this is a plasmid that they put into these cells. What cells did they put in? Was it basically HeLa, the short hairpin TRF2 following ionizing radiation, or TRF2 knockdown? So here they, okay, so here they do show the high. So if you zap them with 5 grays of ionizing radiation, I'm not up on my ionizing radiation units, but I think that's pretty high. 5 grays of ionizing radiation, or they knock down the TRF2, which is part of the sheltering complex at the telomeres. In both cases, you get higher luciferase activity versus untreated, using this plasmid that has the ACE2 receptor driving luciferase. Very promising. Toss up a follow-up experiment. You could probably do some sort of promoter binding assay to see if we have these factors binding at the native ACE2 promoter location, but seems to suggest that that's what's happening. So then the last bit here is okay, how what's the connection between DNA damage, particularly telomere attrition, and the DNA damage response? And then they show in figure 5 that inhibition of the telomeric DNA damage response decreases ACE2 expression, both in cultured cells and in vivo. So let's take a look at that. So here we have, so they're knocking down ATM kinase, which is basically, so add an inhibitor, or they're using short hairpin RNA. What's ATMI? some inhibitor of ATM kinase. Probably look that up, but ATM kinase inhibitor KU60019, some chemical compound. So ATM kinase is required for signaling in response to the DNA damage response. And here they're inducing it by, again, knocking out either TRF2 with the 4-OHT, the tamoxifen treatment, or generating double-stranded breaks using ionizing radiation. Yes, John.
I found an answer to my question. Tamoxifen is used to turn on the TRF2 knockout gene.
It's used to turn on the TRF2.
In other words, to cause the TRF2 to be knocked out, they administer tamoxifen.
Yes, so they're doing that in this paper, yeah, because they have that's the Cree recombinase system that you're referring to, correct?
Yeah, that's on page 4, second column.
Yep. Well, I think you had another question related to that, which is if tamoxifen upregulates ACE2 by itself somehow, right? And we're still, I don't know. I really don't know. maybe it does. So let's take a look at this data here. So all right, so ACE2 messenger RNA levels go up, obviously, after 4 tamoxifen treatment, as we've seen before. The gamma-2A-X foci increase, day 1, 2, 3. This is, so DMSO, and then they add the ATM inhibitor, which should basically blunt the DNA damage response. And the DNA damage response, as a reminder to myself as well, it's due to basically the knocking out of TRF2. And it is blunted significantly, so there's a significant decrease if you knock down ATM kinase, which is basically signaling the DNA damage response. So even though the telomeres are exposed, that signaling, that ATM kinase appears to be at least partially in the pathway, which is upstream of ACE2 transcriptional regulation, because the levels of messenger RNA go down. So they now look at TRF2 mice. So these are, so let's see, how do they knock down those mice again? These are conditional knockout mice. Probably tamoxifen as well, I think. So they do an RT-qPCR detection of ACE2 messenger RNA levels, treated as in A. So this is in mice. And now what they do is they do something really interesting, a little bit different. Instead of using an inhibitor of ATM, they use these antisense RNAs, these anti-telo G, anti-telo C. So 2 different versions that basically target. So these are these antisense RNAs that target these long, non-transcribed, can't remember the prefix, right? So, geez, these long telomeric RNA transcripts that are non-transcribed that come off and from exposed telomeres and are part of signaling this damage response. And you can block them by using these antisense RNAs. So they have 2 versions here, depending, I guess, from which strand they're coming off of. I think they can come off evidently from both strands. So anti-telo G, anti-telo C. And basically the bottom line is, and this is what got the authors excited here at the end, is that you can blunt this response to ACE2 messenger RNA levels by adding in these anti-telo G, anti-telo C oligos to basically, block the signaling specifically at exposed telomeres, right? That are basically, that have TRF2 missing. So these telomeres are exposed. And they show that this is also effective. So this is effective in mice that are so not induced by this TRF2 that's basically conditionally knocked out using tamoxifen system, but also in these generation 3 telomerase double negative mice, you can also block this effect that you see increasing amounts of, so I believe this was cut off, but that's probably ACE2 relative messenger RNA levels also go down using these telomeres. And what do we see here? So here, I believe they look at cell types specifically that are found in the lung epithelia. So these are the AT2 cells. Yep, in AT2 cells. So specifically, because that's where you have most of the concentration of the ACE2 receptor, that also goes down quite significantly when you add in these anti-telo G and anti-telo C. And I don't think, unless it's in the materials and somewhere in the, I don't think they apply these anti-telo G and anti-telo C to the human cells. Not sure why. That's, that would be the next logical step. Maybe they do, but if they did, then, they probably would put that right there. But, considering that this is a very well conserved system in mice and in humans, and that antisense RNAs of this sort have already been used therapeutically, the authors propose that, this is a viable thing to try, probably with some more data that they can, gather as a potential therapeutic to knock down ACE2 receptors, to basically, at least temporarily, in the context of blocking a DNA damage response that's hyperactive, and that might be also leading to increased infectivity of the SARS-CoV-2 virus, particularly in the elderly. So that's where the authors leave it. So I liked it a lot. There's a lot of good to the point data. There's experiments that that I just mentioned that definitely good follow-up experiments that could be done. But I think they kind of laid the red carpet, if you will, for for follow-ups to be done in this direction, particularly with response to the ACE2 receptor and how it's getting upregulated in the DNA damage response. And we could certainly look at senescent cells as well, taken from individuals, and see if because clearly if this is these cells were looked at that were from older people and younger people, but my other question is, well, is the ACE2 particularly enriched in senescent cells in older people? You would think so, right? So maybe it's a subset of AT2 cells from older people, maybe taking senolytics could be a potential therapeutic for a COVID infection if in elderly people, if in putting out ifs here, right? So we're not offering medical advice, for the scientific channel. But if we have an enrichment, I'm just talking out loud here, of AT2 cells that are senescent, and as a consequence of their senescence, they have really jacked up ACE2 levels, and that makes them much more predisposed to infection as a result, right? And then maybe clearing those cells would be a potential therapeutic route. I don't know. Just thinking out loud here.
I think there's a decent chance they are just looking at senescent cells here. Like when I saw the title of this paper, my first thought is, why would like ACE2 get upregulated in response to DNA damage? That's kind of weird. Like, why that? But like in their figure 4, when they show like all the stuff that gets enriched in these cells, a lot of those pathways are things that you expect from senescent cells. Mm-hmm. Figure 4. Oh, I might be. Is that the right one? Yeah, that is the right one. Yeah. So like they've got TNF alpha, they've got the unfolded protein response. I don't know about interleukin 2, but interleukin 6. These are like all things that you get in senescent cells, and I kind of wonder if like an alternative way to interpret these results is that in the context of at least type 2 pneumocytes, maybe ACE2 is part of the SASP, the senescence associated secretory profile. As in that context, a lot of this I think makes more sense. And like throwing 5 grays of radiation at cells, that's the way you can make senescent cells.
Yeah.
And like 53 BP1, that's also something you expect to see in senescent cells. I wouldn't have predicted ACE2 to be like part of SASP, but if it were, that would contextualize a lot of this stuff.
Yeah, or it could be a response to SASP, right? Sort of
right.
You've got the cell because it's having an anti-inflammatory effect supposedly. So you've got SASP coming out, and it's trying its best, right, to anthropomorphize it to dampen that response, right? Otherwise, so it'd be interesting to see if you blocked ACE2 whether or not you got an increase in SASP, right? You would maybe if that's correct, right? And see what happens to SASP if you downregulate ACE2, right? So it appears to be that there might be this kind of, again, there's always trade-offs, right? So elevated ACE2 might be doing a beneficial, might be in consequence to SASP, but then it opens you up to infection, right? Because the virus has basically mutated to take advantage of that, right? So it's sort of a, sort of taking advantage of what might be a potential positive and making it into a negative, right? So, or maybe ACE2 is, part of SASP directly. I don't know. These are questions that are worth following up. Since ACE2 receptor is like, a hugely important molecule for signaling and regulating blood pressure and all kinds of functions. And it's just kind of, obviously it's a big, people in medical community have been looking at this for years, but everybody else is kind of, their eyes are focused on this receptor because of COVID. But it does a lot of other normal functions to maintain homeostasis, right? And that's in general probably what happens to a lot of these factors. They get upregulated. So it'd be interesting to see. I don't know if we looked at a paper, if there's a review paper looking at, again, Steve has always pointed this out with regards to a lot of other factors that we looked at in regards to senescence, that you've got a balance of these factors, and senescence tips the balance. Like some things get a little crazy and other receptors have to get upregulated, or other factors have to get upregulated to try to dampen that, right? So that to keep that homeostatic balance going. So I don't know if we have a useful table out there. We'll probably do. There's probably a review paper which basically looks at things that go haywire, that go up, that increase inflammation, and what are the things that go up in response to that to try to balance that out, right? So so it would be so that way you can kind of tease apart and say that, okay, not everything that goes up as we get older is a bad thing. Those things might be a consequence to dampen the other things that are that are trying to get up and are being damaged, and they're trying to dampen it down, right? So
right, like you might think more ACE2 would be good, right? Because like this should lower blood pressure, like in a very general sense.
Yeah.
Unless it's just like a tepid response to increasing blood pressure. Are there any experiments in particular that you'd like to see as follow-ups to the findings in this paper?
Yeah, besides some of the ones that besides some of the ones that I mentioned, well, I mean, let me just take a look. I think you said there was more. It was likely that those were senescent cells that that they were looking at. Definitely in the context of senescence associated secretory phenotype, right? Because these are ACE2 levels, and they go up in cells that have these, DNA in old mice and in old humans, right? But maybe seeing cells, in the context of what we just spoke about, maybe seeing cells in the context of senescent cells and SASP, and seeing whether or not there's a direct correlation with SASP and ACE2, and then seeing what happens when you knock down ACE2, right? Like if it is a positive, if it is a protective response, if it's a negative response, then knocking down ACE2 would lower SASP. But if it is a protective response, then knocking down ACE2 would potentially lead to maybe not more senescent cells, but worse senescent cells, more SASP, more stuff being secreted, right? So that would be a simple experiment. You could do that completely in vitro. You would just basically take cells that are senescent, stain them with beta-galactosidase, whatever, to show that they're senescent, measure the amount of SASP that's inside the media, right? And then throw in, basically ACE2 inhibitors and knock them down, right? And see what happens, or add in overexpress levels of ACE2 and see what happens to SASP. Now this might be, depending on how the receptor works cellularly, this might be this might require also an in vivo, in situ type of experiment, because maybe it's other cells that will, that maybe the ACE2 receptor is working further upstream, and it's signaling other cells to kind of suppress the SASP response. So maybe you would need to do that in an animal model to see if you then get, cells that are stain less for certain senescence associated secretory phenotype molecules, right? So these are I'm sure if I thought about it a little more, there's probably more experiments I can think of. I mean, that one just comes right to my mind based on a conversation that we just had right now, Michael, about what the role of ACE2 is in senescence and senescent cells, and whether it's whether it's having a negative feedback or a positive feedback response. That's something that should be pretty easily testable, I think, with the right experiments. And then expanding that horizon, right? Looking at all the other molecules that you can then test that way to see if they have an up and down response on the SASP. So that's one group of experiments that in of itself can turn into a paper. But then, all the other little follow-up things, they certainly showed that, that the transcriptional, that the promoter gets activated when they put it on a plasmid. You could certainly do some loose end tying up in experiments and see if you get some of these transcriptional factors that they predicted get upregulated, do they actually bind to the native promoter of ACE2, using a binding assay, chromatin immunoprecipitation assay, or some other related assay to show that that's happening in the native locus. Probably some other stuff if we sat around a little bit longer, come up with stuff. But yeah, I would, maybe there's a bigger experiment that is awaiting in the wings that I haven't thought about yet. But that would be it for now.
Yeah, yours make a lot of sense to me. I mean, one thing that probably the authors wouldn't be so interested in, but after talking about senescence a little bit, I wonder how decent of a marker of senescent cells ACE2 is. Like, I feel like one of the big questions in like the senescence field is what are the decent cell surface markers for senescence? And there've been a few papers publishing markers that are like okay, like UPAR comes to mind. And
in the last paper we did.
Yeah, like this comes up. This is an important topic, and because like if you can find good surface markers for senescent cells, you can like in theory target CAR Ts to them, or just there are ways you can
Mm-hmm.
identify senescent cells easily, and like maybe in the context of these pneumocytes, or if we're real lucky, maybe more generally, ACE2 is a decent surface marker for senescent cells. If that's true, that'd be really handy for folks thinking about like senolytics.
Well, certainly the last paper we talked about, they picked I forgot who the authors were, but they picked beta 2 microglobulin, which was a subunit of MHC1, which is found in every single cell, right? At least these ACE2 receptors are highly enriched in these AT2 cells, and like they're found in other cells, but like really a lot more in these cells that are in the lung. So it's not, this might be a, I don't, but they looked at the ACE2 receptors in the liver too. So it'd be interesting to see if, as a corollary to what you just said, it'd be interesting to see, again, as a marker for senescence, if these ACE2 receptors could, the expression profile could change throughout because that's one thing they didn't do here, I think, unless they did it in the supplemental, do like a, fact sorting or some other system to basically say, okay, I think it was some crazy number, like 1 or 2 out of 43,000 cells like were upregulated for ACE2 normally in like the lung tissue, and it was like these AT2 cells and some other, that were at high levels. But then they go and then they treat these mice with tamoxifen, and they do these deletions of the TRF complex, and they show the levels go up in liver, right, and other cells. So it's like, okay, so the expression profile now is like radical for ACE2 is now changing all over the place. That's more reflective of AT2 in liver cells and other cells that it normally isn't, because these cells are now have telomeres that are short and are senescent. So could it now be, they do go up in AT2 cells, but, are they going up from almost 0 in the other cells to now, well, it's not 0, but, what's the landscape? How does it change? Because it
like what's the fold increase?
Yeah, because obviously brain, you could look at so many, you have over 200 different cell types, right? So is the ACE2 receptor now in senescent cells going up in cells where it normally is not expressed, right? So is it like you're mentioning like a beta, like a galactosidase marker, like you don't see very negligible amounts of ACE2 in the brain, but if the cells are senescent or certain cells are, or whatever cell type you're looking at, adipocytes, right? Does it now go up? Does it skyrocket in senescent cells? And then that paper we did last week or last month, it was only a few weeks ago, I think. That's like, did we do it like, yeah, it was like 2 weeks ago. It was it's, if that's the case, then it would suggest that ACE2 is a better marker than the beta 2 microglobulin, because it seems a little bit more, it would be a little bit, of course we have to test that to see if it's really upregulated mostly in senescent cells, but in that paper they used beta 2 microglobulin as a target for a very toxic therapy that was a senolytic duocarmycin that was bonded to an antibody, and they tested it, and then they said in the paper, like, we need to find other markers, right? So and potentially you mentioned, Michael, that, maybe you could have drugs where you target two markers at once to basically, beta 2M goes up, also ACE2 goes up, right? Maybe a third thing. So you're like, and you have some sort of drug combination where basically the cocktail would be more toxic if it all basically bound to multiple receptors that are all indicative of senescent cell, right? There might be not one magic bullet cell surface marker for senescent cell, but there might be a pattern, if you will, that basically is highly enriched. That pattern is found in 99.9% of senescent cells, but very little in anything else.
I think it's a little weird to me, like with both the MHC1 globulin and ACE2, like these are both surface markers that sometimes aren't surface markers. Like ACE2 gets shed, and M8 that microglobulin also, for unclear reasons, gets shed. So they're like most surface proteins aren't like this, right? But it just so happens that a majority of the senescence markers we've found, or that others have found, are things that like to go systemic. And I don't know if that's a coincidence, but it strikes me as very odd.
It's an interesting observation. I didn't notice that, but yeah, but the beta 2, yeah, that one is like non-covalently bonded. I think it's like that one is just sort of like stuck to the, like, at least from the pictures, right? It's but yeah, a lot of these things, I'm not sure why, if that's the case. Anyway, so I believe this was our first COVID paper. So yay. So good riddance to COVID. But yeah, I think that was a very, very interesting connection because again, we can talk about just general suppression of the immune response and COVID susceptibility, but this paper had a very specific pathway that they basically implicated in COVID entry. And the ACE2 receptor upregulation. Of course, what they didn't do is add in SARS-CoV-2 to show that these cells are now much more likely to be infected by SARS-CoV-2. It's kind of, it was just so obvious that it just occurred to me now. It's like, wait, that didn't actually affect the cells. You would think that would be the case, right? But I don't know, is something wrong with the ACE2 receptors on these cells that you try add SARS-CoV-2 and they can't get infected? Wow, that would be shocking, right? Like that would be like, all right, what's going on here? So I guess, yeah, that would be an obvious follow-up experiment is what is SARS-CoV-2 anyway? Is it BSL-3? Is it BSL-2? Is it BSL-4? What do they consider it? I don't know. But anyway, that would be an obvious experiment to do, right? Basically, or use some sort of deactivated version of it, and probably have a fluorescent payload and see if the infectivity indeed goes up in these cells that are knocked down for TRF2, right? So that would, unless they have that in the supplemental, that would be also like a nice experiment to do. Probably some those and that experiment and some others would probably be necessitated if as the authors wish to have this the antisense oligos being adopted as a therapeutic to knock down ACE2 for these indications. All right, well, cool. Well, I have nothing more to add to this.
Well, you lost for words.
Thank you. May I ask a question not directly related with the discussion, but I heard sometimes one of you was speaking about the fact that aging, process of aging is not always negative. And because of this discussion, is aging a disease? I heard a good argument coming from Ilya Stambler saying, yes, there are some processes of aging who are not bad for our health. And my question is, do you know about one let's say place where, or do you know about somebody who would be having made us kind of a list of things who are related to aging and who are not, let's say, not a process of degradation? I would be interested about this. Was my question clear?
I can't think of anything apart from lower car insurance.
Not well. So just to clarify, Didier, were you talking about I wasn't clear. A list of things that you mean increase in terms of when we age?
So a list of things who would be related to aging, but who would not be bad for our health.
Oh, well, I think that's what I was
like I suppose, for example, white hair. Well, it's not good, but it's bad. You know?
Yeah. You know what? I mean, maybe, I don't know. I'd have to let me Google that for you. It's probably on PubMed. I think, I don't know, but that's that's something that's related to what I mentioned earlier, which is
that's because you all know about this discussion, is aging a disease? And I saw one article from Elias Tambler where he was writing, oh yes, the pathological process of aging. And then I told him, yeah, but all process of aging are always pathological. But he told me, yeah, no, not always. So okay. But
yeah.
If you see something, tell.
Yeah, I mean, certainly, that would fall into I guess the neutral category, right? But
Yeah.
it could just and then a category of things that go up that are potentially beneficial, but then act as a kind of a a negative feedback to any things that are detrimental, that then could be a bad positive feedback, right? So I don't know if there's. I'd be surprised if there wasn't such a list or an attempt at such a list in a review paper. That's something I'm going to look up.
An article about this.
Yeah.
Okay.
Yeah.
What about ears growing, Oliver? Not been ears growing. I don't think there's anything particularly bad about ears continuing growing as you get older, but then like white hair, it's not exactly life-threatening, but it is. The consequences, isn't it? Some go awry.
And
I'm gonna say again, aging perhaps isn't a disease, or as Vadim Gladiev says, aging is not a disease, but it's also not a disease, because he loves to be contentious, doesn't he? Remember, he said that at Erd. But I actually totally appreciate where he's coming from. I've said it before, and I'm going to suggest it again tentatively. It's not a disease, it's a syndrome, right? Which is a collection of symptoms that are often accompanied together, which lead to degradation. It's probably a syndrome more than anything else, right?
Yeah,
Oliver,
it's yeah, my thoughts are, again, in a sense, there's the words, disease, or there's a certain, let's face it, there's a certain arbitrariness to it, and you can pigeonhole things a lot, and you can make a counter argument for it being a disease, because you could say, well, okay, aging affects everyone. Well, but not really, not to the same extent. Somebody who has progeria versus somebody who dies their allotted 80 years and is basically has diseases of aging that basically go up, dementia, or somebody who's a super centenarian and is fantastic, right? So in health, and they're 110 years old. So clearly, there's people that can become super centenarians have, it's not just luck, but there's a certain element of, there's a very strong genetic component there. There's enhanced, I would argue, longevity pathways and mechanisms, right? And so there it's probably not just one particular mutation, right? So you could it's sort of, there's a scale to it. I mean, we call sickle cell anemia a disease, but having one copy is a benefit in a certain environment, right? So there's a certain arbitrariness, and so if you obviously have two copies and you're in the wrong environment, then it can be life-threatening, right? And if you have one copy and you're in the right environment, it doesn't really manifest as badly, and it has a protective effect against malaria, right? And so in this case, aging is a lot more complex, but never mind aging, which is basically, in my opinion and others, basically physical phenomena that are entropic in nature, and every system has longevity mechanisms and longevity responses to counteract that, but to varying degrees and varying abilities, right? So, so the word disease basically is a little too blunt, I think, for a lot of these kind of applications. And it's not a mathematical term; it's a medical term that was derived by consensus, right? So and it sort of fits for certain obvious things, and then you start getting into gray areas of certain combinations of genes that work better in other people than in other people. And so that's my take on it. I think disease is a decent workaday term for medicine. But I think it's saying aging is or is not a disease. It's kind of like shoehorning, something like you mentioned, the syndrome, or something that affects everybody, but to different degrees, into a term that maybe is not adequate. That's my take on it.
So Tuck Finch used to say that aging is the passage of time and senescence is the associated declines with age.
Yeah, I'd say senescence is senescence and related manifestations of of such breakdowns in homeostasis. That's basically entropy in a living system, right? That's how it's manifested, right? And then you've got all these other systems that try to counteract that, the longevity mechanisms. And those mechanisms have evolved in different species, different genera, and different members within the same species to varying degrees, right? And yeah, so I don't, certainly people say, for example, and there's an arbitrariness to it. Certainly people say that Alzheimer's disease is a disease. Not everybody gets dementia. It's Alzheimer's disease. So Alzheimer's is not really, a normal part of aging. I would argue it sort of is, and it isn't. It's part of this syndrome of things breaking down. If somebody made it to the age of 200, perhaps they would eventually, everybody would get dementia, right? If we didn't figure out how to target other things, because
just to interrupt, for me, Alzheimer, or at least neurodegenerative disease, is an example. It's touching almost all, I think, centenarians, most of people who are 90. And so if we could live, let's say, long enough, we would all have, I think, Alzheimer's disease. So for me, it's quite clear. I mean, already the number of people who are 90 years old and who don't have kind of neurodegenerative degradation, it's a very small number. And also, John, I totally agree with. I'm saying that sometimes also aging is the passage of time. Senescence is the disease. Well, the problem is, of course, that we don't have any human being who don't have both together, and that's why people are so afraid of speaking about senescence. And
I feel like we need more words.
And for me, the UN decade of healthy aging, they are really going in the bad direction, speaking about ageism as the problem number one. But okay, digression. Thank you for this small discussion.
No problem. It's wonderful having these more general overview thoughts and discussions on this topic. what was I going to say? Oh, so you know, statistically, right? So it's you know, just to kind of expand the terminology of disease and you know, it's kind of too simplistic to say, well, if it affects everybody, then it's not a disease. Well, I don't know. If you're in an environment where there's a probability, some probability that's not 100%, but not zero, of getting infected with a contagious disease, right? Given enough period of time, everybody, the probability of you being infected approaches 100%, right? So it's a function of time. So does that mean that that virus is not a disease, right? If, you know, like if we use that definitional term. So if we say everybody gets it, within which time period are we talking about? Now, we sort of say that everybody gets it because of, or everybody doesn't, you know, everybody dies of aging because of, you know, well, that everybody eventually dies of some aspect or some component to it. Within their allotted period of time, but you could certainly kind of play that statistical game with a lot of other illnesses, right? So I think we need a better, you know, definitional term of disease.
To go in your direction, Oliver, if we consider that something reaching everybody is not a disease, probably in a very short time, COVID-19 will be no more a disease because everybody will have Omicron.
We've all had a cold, so therefore colds aren't a disease, yeah. Right? So I mean, you know, so it's so that, you know, so obviously you can then kludge the definition a bit and say, well, you know, if it periodically hits somebody and it leaves, then it's still a disease, and if, you know. I just think we need better working definition. Disease is just basically, you know, it's a term, it's a medical term. It works, it's okay for most applications, but it's kind of a kludgy term. You know, once you start, obviously once we apply it to something that we're all interested in, which is aging and longevity, you see that the term kind of breaks down, you know that, and I don't think that's a problem, because as you know, we're looking at it from a scientific perspective, not a medical legal perspective. So whatever the term is, but you know, but I think from a more holistic perspective, it is probably important that these terminologies maybe get a little bit more concrete and based in a little bit more hard statistics, and get clarified a bit, because as much as maybe I don't care as a scientist, the medical establishment certainly cares, and insurance companies care, and the money going into treatment cares, right? Because because it's all dependent on how these things are pigeonholed with terminology, right? And if there is no box for it, it just gets ignored. I mean, it's like, well, it's like aging. It's like, let's take a look at the list of things that we can check off for money. Nope, it's not on there. Sorry, we don't. You know, it's like so in that respect, it's a problem, right? So if we can't integrate it into the kind of medical establishment, scientifically, it's not a problem. But for operational medicine, it probably is.
It's also not a clinical endpoint as well, I wanted to add. So age reversal is not recognized by the FDA in particular as a relevant endpoint for clinical trials. So they're looking for disease modification. So yeah, again, that's why I think the merit in our field would be the idea to work on disease modification. You can use all these rejuvenation techniques, but at the end of the day, that's what the FDA cares about, is it's better than the current standard of care, and it also is demonstrable of disease modification. So that's what I think we should really sort of focus on, because we are not going to win that battle about aging is a disease with them. We will die on that hill. That's what I think anyway.
Yeah. So, there's certainly, from a medical perspective, I see a couple of things on the horizon. One is, like you mentioned, Steve, like looking at it from a syndrome angle, where you basically just arbitrarily say, okay, the majority of people are going to have this cluster of chronic diseases, right? And those are the ones that correlate with a lot of aging manifestations, right?
Well, your hallmarks, Oliver. I mean,
Yeah.
your basic standard 9 hallmarks of aging,
Yeah.
or your 7 deadly things, whichever you prefer.
Yeah.
They are your syndromes. There's your syndromes right there.
Yeah, but I'm talking more that the medical establishment
symptoms.
will recognize. They're not going to recognize shortened telomeres, or they're not going to recognize the Horvath clock, because this is too still hypothetical. That's something that will get accepted once you know. So I think that's one front from the, you know, like diagnostics that are at the molecular level, but then more kind of proxy measures that are chronic diseases of aging, because chronic diseases of aging, such as, you know, obviously various dementias, various cardiovascular diseases, various types of cancers, you know, a lot of these things cluster with an, you know, age demographic, and you're seeing you know, some studies right now looking at several of these manifestations being used as kind of a proxy for aging, right?
We could add sarcopenia is another.
Yeah, idiopathic pulmonary fibrosis was used, and some other, you know, so if there could, you know, so I, you know, that's something that's kind of a low hanging fruit for you know, medical trials. You know, that's something that is already, the boxes are already there. And you can easily imagine a clinical trial being used to treat a cluster of these things, right? Where we know that really it's tackling the root causes of aging, or you know, either dampening that or boosting a longevity mechanism, right? But as a consequence, you're really, you know, the medication is saying, you know, it'll treat these 3 things or these 4 things, right? And you know, so I think that's, you know, that's certainly the direction that we're I think seeing things move. You know, this radical redefining of things will happen later, as paradigms shift, and as, you know, as aging creeps up with the people that are holding back progress, right? Who said that before? That, you know, that science progresses when the folks that basically hold back the new advances die off, right?
Yeah, progress is measured by the obituary column of scientists, and other leaders or experts. But, you know, if you want to expedite that, just get me some names and don't ask me questions, and I'll see what I can do.
You know what this means, Steve? That if we actually do defeat aging, that scientific advances will be made via trial by combat.
Trial by combat, and I'm just going to tentatively suggest to satisfy the geeks out there, the geekiness. I'm going to say that the chosen weapons should be lurpers, as seen in the classic Star Trek episode where Spock and Kirk fight. A lurper being a kind of cross between a traffic bollard and, what, as far as I can tell, a lawn edger. I don't know what the hell they are. But they look cool, so let's do that.
I guess be careful what we wish for, right? Because it's like after the end of the Cold War, the nuclear threat sort of subsided, and then you had all these hot little conflicts, and it's like, hey, we defeated aging. Bad news: 100 years from now, all the Nobel laureates from the sciences are all basically buff mercenaries, because that's the only because that's the only way you can finally convince, you know, your colleagues that you're right. Because you have to actively kill them off in trial.
Yeah, but ultimately that'll lead to one ruling over all the others and rising to the top. And that's how you get Khan, you know. I'm just saying, guys.
Seriously and very short, I'm absolutely not convinced by this famous thing, because, for example, in the IT field, well, people had no time to die, and there is acceleration very clear, and so on. So, people were never living as long as now, and we cannot say that we are in a period of, let's say, decreasing scientific progress. Well, even if I think that there is kind of a decreasing scientific progress because of patents and IT and so on, but that's another question.
Yeah, no, I agree with you, Didier. It's
Yeah.
a tongue in cheek thing. You know, I think also a lot of advances in science, like in molecular biology, come from barbarians coming from other fields and storming the gates. Like you had a lot of physicists coming in and basically quantifying molecular biology and basically and changing how experiments are done. So you know, you have a lot of that happening. But yeah, so bringing that back, the Christmas season is almost here. That means Festivus is almost here. So try feats of strength around the Festivus pole. So you know,
or what is it? Kwanzaa?
Yeah.
What's
Yeah.
that? That happens around this time of year as well, I think.
I think it's a
Kwanzaa.
day. Yeah, the 26th. Yeah.
Yeah, but there's no
So,
airing of grievances for Kwanzaa. It's not as good.
Before we stop, can I ask also one meta question? So
Yes.
this is interesting, small discussion and so, but why don't you try to have more people, or how many people receive an invitation here, more or less?
I would like to invite everybody. I don't know, Steve.
they're the rules,
I think-
Oliver. I'm not allowed to invite everyone because
Only the
everyone-
people giving money, I think, eh? Something like that, no?
Didier, you
No, I'm not
you-
joking. I mean, the people who were giving money at one time, no? I thought so.
yeah, we've got it's actually a two level system. People who support us donate and support the organization, they get to be live on the call like we are right now, but we also are live streaming, of course, to Facebook right now. And
Okay,
people
I
can
didn't know
also
that.
you can also talk to us in the chat and ask questions. So
Okay.
we feel that's about the best way to deal with it. And obviously everyone then does get to join in. I mean, I would like to invite everyone on, but
Okay, thank you for the information. Yeah, it's like many things. I'm always a little bit disappointed that I see always the same faces. Well, I'm happy to see always the same faces, but I would like to see more faces. I mean,
Maybe we can invite everyone, and if you donate, then you can be unmuted.
Oliver, no, but I do actually have some news along those lines, Didier, to share. We are actually next year we're going to start doing more live webinars or things like that, like live events, and that's going to be a lot bigger with more people able to just join us. I'm not sure what on the pricing. Hopefully it'll just be everyone can join in. It'll be a free event. But we're going to do a series of those next year, and we're thinking about things like discussion panels between experts and debates, because I'd like to see some sort of actual debates. Yes, I'm going out there. I'm going to say program versus random damage arguments fight. It could be great, man. It could be great.
Well, not too many fights, but yeah.
Yeah, we have that fight in the Discord.
Robust. Should we say robust discussion?
I think we had enough fights about somebody that you probably know. The last months, it was enough, and it's not finished. And okay, I have to go now. Great to see you, and to really lifespan.io is something absolutely great.
Thank you.
I maybe my last sentence, but that's also for another time. It would be great if it was possible to have kind of lifespan.io smaller in other languages. That's one of the things I'm thinking about one day when I will have time. But I'm a little bit busy.
So we are. I'm just going to say that we will be offering articles in multiple languages. I think it's about 47 languages. We're in the process of actually implementing some language plugins, which allow which will allow it to be translated and voiced as well. So you'll be able to hear it in your preferred languages.
DeepL is now almost perfect for language. Google Translate a little bit better, a little bit less good. Yeah.
Yeah, it's not so good at Russian.
I will not need to live 1 century more to be able to speak English correctly. Yeah. Okay.
So take care, Didier. So
Goodbye.
so we can have a spin-off like Lifespan X, not related to TEDx.
Yeah.
Right. Pronounced
Bye.
lifespan X, not Lifespanx.
Oh yes, yeah. Oh God, don't start that again. We had
Oh,
that a
start?
couple of years ago. Yeah, well, this was a thing a couple of years ago. Never mind, I'll tell you later. But
Okay.
yeah, we did have the whole Lifespanx thing discussion, and
Jeez.
I did mention it 2 years ago.
Oh God, I don't remember that. Maybe that was in the back of my head and just like circled. And I thought I had just made something up.
I was thinking, what kind of organization am I donating to? No, we're not that kind of organization. So anyway, on that not festive note,
Okay.
hopefully everybody has a good, festive season. However you celebrate it, if you celebrate it, if not, you do you. Enjoy yourself, stay safe. We will return next year. Sounds a long time away, but it's not.
Yeah.
And
Very soon. And don't drink too much eggnog around the Festivus pole because you might, injure yourself. But other than that, have fun, stay merry, stay warm, and stay safe.
Yeah,
Thank you, Oliver, for leading this discussion.
Oh, thank you all for attending. And see you all next year.
It's not that far away, Oliver. All right, guys. Thanks for joining us. And if you are interested in joining us live on the calls in future and getting involved, do check out the Lifespan Heroes. You can do that by visiting our website at lifespan.io/heroes. We will be in touch with the new programs, and hopefully next year we'll be also announcing those new live stream events, which should be a little bit more robust discussion. No fighting. We won't have any fighting. Don't worry. I'll break it up. But we'll see you next year, guys. And stay good. Make sure you don't end up on the naughty list.